400 years after scientists first discovered red blood cells, we're still uncovering fresh surprises. And the latest might just change how we think about blood clots. Turns out, red blood cells don't just ride along during clot formation. They actively help your blood clot tighten and stabilize. For centuries, only platelets were thought to do the heavy lifting when blood clots tighten. Now, researchers at the University of Pennsylvania are rewriting that story. In experiments, clots engineered without platelets, or with platelet activity chemically blocked, still shrank by over 20%. That's when the scientists thought, red blood cells must have more to do with it. So what's behind this? Enter osmotic depletion. Here's the sci-fi sounding scoop. Within the fibrin network of a clot, red blood cells get packed tightly. Proteins outside the cells, now crowded, create higher concentration on the outside than between the cells. This imbalance generates pressure that pushes the red blood cells together, helping the clot shrink, even without platelets. The team also tested for another mechanism, protein bridging on cell surfaces, but found it plays a much smaller role compared to osmotic depletion. As Rustem Litvinov put it, this discovery reshapes how we understand one of the body's most vital processes. And it's not just trivia. Understanding how red blood cells contract clots may lead to new strategies for tackling bleeding disorders or dangerous blockages like strokes. Imagine treating patients with low platelet counts, or finding new ways to prevent strokes caused by traveling clot shards or embolisms. This research paves the path for strategies that go beyond targeting platelets alone. So next time you think of RBCs, remember, they're not just oxygen couriers. They're key players in the mechanics of healing. If you're curious about more mind-blowing science discoveries, hit subscribe and let's explore together.